Okay, well, I'm going to go ahead and get started because we have a busy day, and I want to first thank you all for coming. This was, you'll hear from Pascal how this meeting got sorted out, how we organized this meeting, and then we'll hear sort of a lot of history about science, of new things, old things, and not only about your field, but also about UCSD and the various ways that Palmer's contributed at many different levels. And I just want to thank everybody for coming. We were stunned that we got such a big response, so many people coming from all over the world. And it's like, you know, I think at this time of science where we're all kind of stunned by what's happening, we've sort of lived in a golden age where we could meet around the world, we could share our science, we could spend our lives doing science. It's really been a golden age for scientists, and I hope we restore something that that our children and our grandchildren can do some of these things. But we go from like thinking about molecules, and so I just wanted to say, like, the first acetylcholinesterase meeting was in 1975, I think, in Split. And so it shows we got to learn about all around the world. You guys have met all around the world, and so science is a world language. And so it's been 50 years since that meeting, and the first structures, the first acetylcholinesterase structure was in 1991, and then we began to think about not only the molecules, the chemistry, but also these molecules, how do they work? And Joel and I realized at the last meeting we had in Slovenia that his first paper of acetylcholinesterase and my first paper of protein kinase A were in the same volume of Science in 1991. So anyway. And we're going to hear them at the end of the day today from Ryan, who's shown us how far the field has come. But I think what's unusual about all of you is we're not just structural biologists, we're not just a pharmacologist. Ryan knows physiology, Ryan knows pharmacology, and he also knows structural biology, and it's really putting all this together that's how we can finally figure out how this works. So I'm going to turn it over now to Pascal. Pascal should be sitting down here too. But
should do the reverse. So good morning, everyone. And it is a great pride and privilege, and I think with a kind of a tremolo in my voice today, that on behalf of the International Steering Committee, I welcome you on this very special symposium aimed at honoring Palmer Taylor as a scientist, scholar, mentor, department chair, and dean, as had been so nicely formulated as the previous symposium, which was held here in 2016. Many of you were here 9 years ago. Some others were not, and we are delighted to have you here today. Also, let's have a thought for those participants who couldn't come to join us this year, and which we all regret. The local organizers of this 2025 symposium set up a very beautiful website as an extension of the one that was created for the 2016 symposium. It is easy to reach with simple keywords, Palmer Taylor Symposium. And here you can find everything. I mean, the program, and I can show you how it looks like. Here it is. Short bios of the speakers, list of the confirmed participants, information about location, transportation, parking options near the SSPPS building, and here is a nice slide made by Zoran. Information about time and direction for the dinner of tonight at the Birch Aquarium. And we have still another one. Okay. And of course, the many, many, many photos that you sent, and which will also be on display tonight during the dinner. This symposium will take place over 2 days, I mean today and tomorrow, and we will have 30 speakers Among them, 11 traveled from far away, either clockwise or counterclockwise, and they may well be suffering from jet lag. One came from New Zealand. Here it is. One from China, 2 from Japan, 1 from Israel, maybe several of them also, 2 from Poland, 1 from Germany, 1 from Croatia, 2 from France, and we have a third French speaker who will join us by video means, and who is here, over there, in principle with no jet lag. Okay. Some other speakers traveled from other states of the US, so Maryland, Michigan, Texas, Utah, or from other places of California. Still others more simply come from the other side of the street, or even down the stair of the SSPPS building. So beside the speakers, this worldwide distribution holds for several of you in the audience. So whatever your role at this symposium and whatever the distance you have traveled to come here, thank you all for being here today. And dear Palmer, among the 120 participants of this symposium, you will recognize many of your former students, postdocs, staff members, visitors all along the years, many colleagues and collaborators, many friends. And I think that we can say that all of them are true Palmer-Taylor aficionados. Okay? In fact, the final program of these 2 days differs much from our initial plan, which were more concentrated on former members of the Palmer-Taylor Lab, 1 single day at a much smaller conference room. But it happened that for each of its meetings, the steering committee became more and more inventive and enthusiastic, so that not only did the program gradually evolve into what it is today, but we also had to restrict our imagination so as not to include a 3rd day and keep you busy on Sunday as well. Since money is never an option, the steering committee warmly acknowledges financial support granted by the UC San Diego Schkack School of Pharmacy and Pharmaceutical Sciences and its School of Medicine, Department of Pharmacology, by your kind personal donations, and by Amgen. Okay? This support enables us to use this very nice auditorium and its technical facilities, along with assistance from the local staff. To enjoy morning coffee and bagels, which I hope you got, before coming here, coffee breaks, lunch on site for 2 days, and to celebrate Palmer further with the dinner of tonight at the Birch Aquarium, where the Taylor family will join us. Talking about food, your badge holder contains 2 kinds of raffle tickets, a red one, 2 red one and a green one. Okay? To obtain lunch today and tomorrow, you will have to present a red ticket. While to enter the aquarium tonight, you will have to present a green ticket. So make sure not to losing them, because otherwise you would get nothing to eat. Now let's turn to some other practical information. Speakers. You received instructions on when, how, and to whom, you should provide your presentation and the format to use. So hope you can comply with them. Also, please do your best to keep on your allotted time. Moderators, we have a tight schedule, so please do not duplicate the speakers' bios that are posted on the symposium website. Also, note that the allotted slots are of varying lengths. So check twice in order to manage time at best, okay? And for my part, I have to confess that I'm usually much better at respecting time limits as a moderator than as a speaker. So my moderator will have a lot of work with me. Okay. Group photo. There will be a first shooting today, just before lunch, here in the auditorium, with everyone standing on successive rows because of the bad weather of today. So please do not run away after the last morning presentation. should the weather permit, I mean, we will make another photo outdoors later today or tomorrow. Fingers crossed. Everyone, internet connection in the auditorium is through logging UCSD-guest, like everywhere on the campus. Again, information on where to park near the SSP building and driving directions to the Birch Aquarium for tonight's dinner are posted on the symposium website. Driving directions to the Taylor's residence for tomorrow's dinner will be provided to guests as a printed leaflet. Tonight's dinner. You received a call for personal photos, videos, anecdotes from your time with Palmer. I hope you were able to gather many of them and are willing to share them with all of us during the dinner. Whenever you are ready, just come to the computer with your USB key stick and tell us your story tonight at the dinner. Finally, I am sure you will agree that the steering committee also deserves praise, with special thanks to its local members and their associated helpers who took care of the logistics and all those numerous details to consider and manage. But perhaps, I mean, you want to check how it is. I mean, it works first. I don't know. We'll tell later. So at that point, I think we can announce that the Honoring Palmer Taylor Symposium is open. Welcome to everyone.
How do I make it go for it? Put mine.
Hiding behind.
I can go for it. This one.
Yeah, yeah, yeah. Okay.
Okay. Okay, so Palmer and I wanted to say a few words, but I was going to take a few minutes to introduce you to Palmer growing up and some of the things maybe you don't know about Palmer. So Palmer comes from, let's see, does it go this one? Okay, so Palmer grew up from a long history of pharmacists, and this is a picture, some of you, it's in the lobby there. It's a picture of the Taylor Brothers drugstore that was started in Stevens Point, Wisconsin in 1896. So that was his grandfather that started that. And so if you go back and look at I'm sorry, it's like, here, I can put it here now. Now it's working. Okay, so this is Palmer's grandfather when he was a young man in the pharmacy in Stevens Point, and probably about 1914, 1915. So his father was a pharmacist. But as I went back and looked at some of this, I knew his father was a pharmacist. That's his grandfather, William Wallace Taylor. He was a pharmacist. He came from Ireland in the mid-1800s and then ended up in Stevens Point. So he was a pharmacist, so I knew that. But it goes even farther back than that. So it's his father it was a pharmacist. His father was a pharmacist. His grandfather was a pharmacist. His grandfather's uncle was a pharmacist who actually then went back to medical school and ended up as an obstetrician and was on the faculty at UCSF, actually, in the early 1900s. Then his great-grandfather, William Potter Palmer, was a pharmacist in Sparta, Wisconsin. And then what I learned, his great-grandfather's father, Hageman Palmer, was also a pharmacist. So he had like pharmacy in his blood for the longest time. And this is also, this is Potter Palmer, the Potter Palmer family. That's the same Palmer family. It's a very famous family that came from three brothers came in 1650s to Connecticut, and they got the Palmer House, Potter Palmer in Chicago, this branch, and then a branch that went up to Detroit with the Kellogg family. Anyway, it's a pretty interesting family. I'll learn more about that. But then he came down. Palmer was always very smart. He was valedictorian of his class. He remembers like all of the teachers he had since kindergarten, and so he likes to lecture. But he's very quiet, and he was not really a leader. He came down to Wisconsin, and I'm going to tell you this next story because I think it exemplifies how some of the most important things in our lives happen because we never planned them. They're things that never happen. He's in line, even then, back then, there were long lines for freshman registration for classes. The crew coach came up to Palmer and asked him if he wanted to go out for a crew. And, crew coach was looking for these big guys. He came from Washington, the coach, and big guys, strong guys, these dairy farmers who'd never heard of farm crew, but they were strong. And then he came up to Palmer, and I don't know why he approached Palmer, asked him if he wanted to go to crew, and if he signed up for crew, he could get his labs in the morning instead of in the afternoon. And so Palmer said, oh, that sounds good. And so Palmer actually, this was a really formative, if I ever write a book about Palmer, this will be how I think Palmer learned to be a leader, because he was coxswain of this crew. And they went on to win in 1959, when he was a junior, they went on to win the national championship. They tried out for the Olympics. They didn't get to the Olympics. But these are some of his best friends. Several of these guys were in our wedding. He still talks to them periodically. This was a really, and what he learned, maybe some of you saw or read Boys in the Boat. And the thing about a crew is that it's not one guy. You have to have all strong rowers and a really smart coxswain. But you have to work together. It's a team. And so I think being on this crew is what taught Palmer to be a real leader. And that's what he brought here. I think many of you know that when he built the pharmacology department, the school, it's like everybody working together as a team. That's a really important message. And that's kind of governed his life. So then he went to Wisconsin to learn chemistry and pharmacy. And then he went to the NIH, and Larry will tell you a little bit more about that. And I think at NIH he learned physiology and pharmacology. And then he went to Cambridge, and there were two people who really were a big influence on his future career. Arnold Burgeon, who was head of this new molecular pharmacology unit, that's thinking about the molecules, that not just the drugs, but the molecules. And then he actually spent three months in Manfred Eigen's lab, where he learned T-jump, temperature jump. And then he actually met Jean-Pierre for the first time. We just realized this last weekend when we were talking with Jean-Pierre in 1969 at a meeting in Finland. And Jean-Pierre had just come back to Paris, was doing his historical Alistair studies, and then Palmer was just starting to think about what he was going to do. And acetylcholine is what he decided, that he was going to work on acetylcholine, and so that became the molecule that kind of defined his career. And, mine is cyclic AMP, his is acetylcholine. And so I think what he learned in these early years was chemical pharmacology, sort of classical pharmacology, but then what really intrigued him was Arnold Burgeon's vision of molecular pharmacology. What do these molecules look like? And that's really driven him in his career. And this is what the basic science building looked like in 1969. This is a new school. We started in 1965. You can see in the back, there's Muir. Here. That's Rebel College, and I think there's Muir, but there's nothing in between. There was nothing else on the campus. This library and that's all that was there. And then if you go, that was when they moved in 1970, and so Palmer was the second person to be hired. You'll learn that also from Larry. They moved in on the second floor along with the physiologists, surrounded by physiologists, part of the division of medicine, and I actually moved into this building on the fourth floor. The top floor. I was up there. I came as a postdoc. I was there with Nate Kaplan, Jack Dixon, Craig Venture. Craig will be there this evening or later on. Craig was a graduate student. So this building, there's nothing else around us. It was just empty. And you can see how much it's grown in all this time. And so this is Shelley, and Shelley is down here. This is in the early, she was his technician in those early years, and that's Palmer and Shelley working in the lab. And she was with him for such a long time and worked with so many of you. And then this is that sort of motley-looking crew that Palmer assembled as the Division of Pharmacology, a division of the Department of Medicine. And you could see, like, a lot of these people are here today. I mean, it's really pretty amazing. Vince Dion is here. Mort Prince is not here. Palmer, and then Paul Insel is there with his famous beard. I wasn't pharmacology there. I was kind of the mother of the pharmacology department. There's Steve Mayer, who was the chief of the division. Larry is there. Harvey was here. Joan Brown. Joan Cantor wasn't actually in the department, but she was part of this team in the early, probably 1978, 1979. So this was the group, and I think what's interesting about this group is that these are not people working on acetylcholinesterase and Palmer's field. They're people working on molecules, it's molecular pharmacology and molecular physiology. You have cardiologists. You have Paul is a clinical pharmacologist. You had hypertension with Mort. You were thinking about the physiology as well as the pharmacology, as well as the drugs, and as well about the molecules. So I'm going to let Larry take over from here.
Oh.
Palmer.
Oh, okay.
I said I never turned off my phone. Nobody calls me except when you don't want them to, right? Just the text.
All right. So we close that.
Is this the first slide?
You can use this to advance, and this is pointer.
This is the,
Yeah, this is laser pointer. You can use this to advance slide.
Maybe you can push the button. Maybe you can forward the slide.
Or with this.
okay,
Let's see.
let me try that.
Oh, it's not working.
Let's see, is that the first slide?
That's the first one. Oh, no, this is number 4.
Yeah, there.
This is your hope.
Okay, and then I push it.
Yeah. Let me see.
Okay,
Yeah, this.
okay,
All right. So,
very good. So, here is a synapse, which the blue is the presynaptic and the green is the postsynaptic, and the two vital molecules that we had worked at in both the central nervous system and the peripheral nervous system were the acetylcholine receptors that were present and acetylcholinesterase. And both of these working together in very close proximity gave rise to the rapid end plate potentials and disappearance of the end plate potentials by the cholinesterase sopping up the release of the vesicles of acetylcholine. There were other molecules in the synapse that we also worked on, and these are synaptic adhesion proteins that you can see right here. Norexin is presynaptic, neuroligin is postsynaptic, and they form matches in different terminals to distinguish terminals, and allow these synaptic vesicles to be released onto the receptor for very short periods of time and then be scooped up by the cholinesterase. And so we learn about these two molecules, the cholinesterase that's shown in the corner there, let's see, there, which can be present as a soluble tetramer. It can be a link to a membrane form. It can be linked to collagen in the neuromuscular junction as well, and looked at the adhesion molecules that bring cholinesterase in the vicinity of the receptor. And then our second molecule here was the acetylcholine receptor, which, what happened here?
Better go back.
Yeah, the acetylcholine receptor here, which is a transmembrane protein, all of that, and the extracellular portion is shown in the bluish color there, and that's where the ligand interacts. And then there's a channel that goes across the membrane in the gray portion there, and that's how it becomes a transmembrane channel activated by the molecule acetylcholine. So we worked with all three of these entities here, as shown in the next slide. Here, the cholinesterase, and you can see a group of people, most of them are here today, working on cholinesterase, was the first molecule that we worked on, and then went on to the nicotinic acetylcholine receptor as a neighboring molecule, and you can see a number of people here that worked on that are already here today, and various avenues of that extend across our campus, extend into Europe, extend into Japan, and even into New Zealand now. And then when we go to Neurexin and Neuroligin, Neurexin being the presynaptic, Neuroligin being the postsynaptic, we have another set of individuals that have worked with that as well. And you'll see some overlapping names, like Shelley Camp, that had worked in many of these different areas as well. And so this then becomes the compasses of our laboratory over this period of time. Now, the purpose of my being here is not to talk to you, but to learn about many of the things that you've done since you've come from San Diego. And therefore, I will keep my talk short, but to mention to you this developments that occurred in my training. And as Susan mentioned, it was a matter of going from chemical pharmacology at the NIH, which had to do with the nerve endings, into molecular pharmacology with Arnold Bergen at the University of Cambridge. And I learned about what he was doing of coming from McGill University in Canada. And he was going to come and do molecular pharmacology and look at molecules instead of systems. And that's how they developed then these two training areas of NIH and the University of Cambridge before I went to a more modern place like UCSD. So with that in mind, I traveled throughout Europe, worked with Manfred Eigren and Göttingen in Germany and Cambridge, went back and forth between those two locations because Cambridge had the fast reactions for stop flow kinetics. Göttingen had the fast reactions for equilibrium relaxation or temperature jump work, and we worked back and forth between those locations before coming to San Diego. Now, another part of life with me was what was happening with my wife, future wife, Susan, who was in Cambridge, and we went back and forth, and you can see from Susan from graduation from high school, meeting me, to part of the becoming a scientist, looking more like a scientist as the years went on. So with all regards to that, you can see that development occurring over a number of years. So with that in mind, that's a number of things have happened with our family. Ashton is working in the computer industry downtown. Tasha is practicing pediatrics here in Chicago. They have, and we have the 5 grandchildren here, and get them all together typically at Cranberry Lake up in upstate New York, which Susan's family had, and that becomes the other half of life here as we not only do science but reproduce as well. So with that in mind, I'll thank all of you for coming, and I'm absolutely amazed at the attendance and the age span. I think this is the first example of a large gray-haired audience coming into the School of Pharmacy, and we welcome you.
This one? Yeah, or try. Yeah, I think this will work. Just back and forth and point there.
I'm Larry Brunton. I have never worked on acetylcholinesterase. I have owned a bottle of acetylcholine, and I'm familiar with Palmer's work. And I've been asked as a emeritus member of the pharmacology department to review Palmer's time here from 1970 to 2002. What sort of place did Palmer come to? What was happening on this mesa in 1970 when he came by sort of on a chance meeting to interview a story that we'll tell tonight? Well, there wasn't much here. The Salk Institute was fairly new and doing well. The Scripps Institute of Oceanography was the core, the center from which UCSD expanded and became a university. But the campus itself was rather small, only about 6,000 students in 1970. The medical school, well, what was happening there? It was very new. Only 47 students in the first class, which was a group that entered in 1968. It was very oddly organized. There were no basic science departments. That created quite a stir nationally, among scientists at least. And the basic scientists were assigned to be members of clinical divisions of clinical departments. And the division of pharmacology was going to be in the Department of Medicine. Now, the people who founded the Department of Medicine were quite a notable group. Gene Braunwald, who's still with us and emeritus professor at Harvard, was the first chair of medicine. He brought with him from NIH a number of people, so many, John Ross, J. Siegmiller, Dan Steinberg, that they referred to themselves as NIH West. All of these people were well-known scientists at the time. John West, coming from Australia via the UK, was arriving just as Palmer was here interviewing. And these divisions were all strong divisions. The leader of the Division of Pharmacology was, well, the division was small. In fact, it was just Steve Mayer, the division chief, and Steve had the good sense to hire Palmer, who came by at the end of a long U.S. tour. This was his last interview. And what Palmer did was interesting. He talked to Steve, and they hit it off. Steve had come from Emory. He raised quite a ruckus in the Society of Pharmacology because he chose to enter a medical school where pharmacology would not be a department. He was a close friend of Ed Krebs and Eddie Fisher. He was interested in transmembrane signaling and the consequences. He was very skeptical that cyclic AMP could play as many roles as it seemed to be playing. He especially thought maybe it wasn't so related to the changes in cardiac contractility and cardiac rate. And he was very active in the American Society for Pharmacology and Experimental Therapeutics, our professional society. He recruited Palmer, and then there were two people in this division, and they were teaching all of pharmacology to the medical students. So there was a very busy few years before other people were added to the department. The two amazing things about Palmer's recruitment: he did not give a seminar. He just came, he and Steve chatted, Gene Brownwall dropped by, they all agreed, and Palmer was hired. The second thing was and you won't believe this, but it's true, he accepted this job before he talked to Susan about it. So she was quite surprised. She wasn't quite sure where La Jolla was. And he phoned her in Britain and said, well, we're moving to Southern California. And they're still married. Now, the faculty recruitments during the Mayer era are these. Mort Prince came on 2 years later. Two associates of Steve through graduate school and postdoctoral studies, Steve Gross and Jim Stoll, came in. They didn't stay too long, but the department began to grow with the addition of Paul Insel, who brought clinical aspects but was very interested in cyclic AMP. Vince Dione, who's here also, as is Paul. Vince set up patch clamping. Chaim Leffert was very interested in liver regenerative properties and really got us all thinking not only about matters of transport, but about matters of repeat sequences in DNA. And Joan Heller Brown had been working here with Steve as a postdoc on a junior temporary faculty sort of position, and she joined the faculty fully with a grant and her own program in 1980. So these were the recruitments during the Mayer era. Now, it was a happy bunch. We met once a year at the Federation meetings, usually a big meeting of all the biology groups, and we got together frequently if the meeting was in Anaheim. You can see Palmer spoke at every one of these social gatherings, and people were smiling. They were happy. Palmer always had a good way with a joke, with a demonstration. There was also a beer chugging contest, and Palmer could win it every time. He persuaded the group that he could open all the sphincters in the esophagus, which most people cannot do. Well, there was a secret to that. May I tell the secret, Palmer, or shall we keep it a secret?
No, you can tell them.
The secret was there were two identical glasses, but one had a lot of glass inside and a smaller volume of liquid. And Palmer got that one. They looked the same when the beer was added, but he was drinking only a third as much as whoever the competition was. And he won time after time after time, and he could do it again. Now, as Steve Mayer, Steve had some health problems, and as his health declined, Palmer played a greater and greater role. And one of the things that he did that I think was most important was he hired me. I'd been a postdoc here with Steve. I was interested in cyclic nucleotide actions, doing work both on the heart and in various cultured cell systems. And so I was appointed in two divisions of medicine, pharmacology and cardiology. And you'll see that this is a theme that Palmer and Susan have both emphasized, is moving across disciplines and filling people into appointments in multiple departments and creating some really cross scientific interactions. Bob Tukey came, a toxicologist, and was jointly appointed. And then the department started to grow. In 1987, under Palmer's leadership, he was now the official head of pharmacology division. The division became the first basic science department in this medical school. And under Palmer's tutelage, we hired Mike Caron, Jim Farmisco, Tony Yaksh, Roger Chen. You'll remember Roger, I'm sure, a Nobel laureate who taught us how to make and use fluorescent probes to find out what goes on inside of cells in real time. And a lot of that work continues with Roger's trainees here. Joe Adams, expert in kinases. Gerald Chun, who really invented the field of lysophospholipid signaling. Annie McCammon, who will speak later today, who made great leaps and taught a lot of us the value of computational analysis, looking at molecules and predicting how they move and breathe, finding new nooks and crannies where ligands might bind. Alexandra Newton, the world's expert in PKC. Phil Bourne, who came to manage the National Protein Data Bank and made that a very useful thing before going on to work at NIH and now at the University of Virginia. So it was a wonderful group of people that we had and were hired under Palmer. Now, there are other feathers in Palmer's cap during the time when he was chair of pharmacology. There's a training grant, the UCSD Pharmacology Training Grant. It's been funded for 45 years, and it's been the centerpiece of funding for our graduate program. Palmer was important in the early days of the formation of the physiology pharmacology PhD program, which really happened between some cardiologists, John West in pulmonary physiology, and the pharmacology department, and Steve Mayer and Palmer. That's now our biomedical sciences program, but pharmacology continues to play a central role in that program. Steve had been the editor of one of the leading pharmacology journals, Molecular Pharmacology. It was still a fairly new journal at that time, founded at Stanford by Avram Goldstein. And Palmer was the associate editor at that time when Steve was the editor. Palmer went on to become the president of the Pharmacology Society in 1995. That was a funny time for the Taylors because Susan was president of the Biochemistry Society, and they would meet in Chicago airports and otherwise often didn't see each other for a week at a time. They were so busy flying back and forth. And then the feather that rises high in his cap is the founding of the School of Pharmacy and Pharmaceutical Sciences. Palmer got the seed money from the Skaggs family. We wouldn't be sitting in this room without Palmer and that seed money. This building wouldn't exist. The school has really prospered. Palmer was able to co-opt those of us in pharmacology to do some of the lecturing at first, and now the faculty in pharmacy is strong. It's been a good thing for the school. So I will leave it there and tell you that Joan Heller Brown became the chair of pharmacology as Palmer ascended to deanship, and Joan's going to talk about our pharmacology department since 2002 when she took over. We probably ought to say hello to Jean-Pierre. How about a bonjour Jean-Pierre on 3? 1, 2, 3. Bonjour Jean-Pierre! Did that wake him up?
And this is the change. Forward. Okay. Thank you for having me talk, and welcome to all of you. It's great to see these people here. I realize that
what I'm going to say at the beginning probably applies to many of you, and that is that it would be no exaggeration to say that Palmer made my career. And while I'm going to talk about the department and its development, as Larry asked me to do, I want to emphasize how important Palmer was to me, as I know he was to many of you. So, and most of my slides have already been shown, and the concepts introduced, so I can go through this quickly. But I came to UCSD in 1975 as what was called a research assistant professor, and this fellow here, Steve Mayer, was head of the Division of Pharmacology, and I think he looks a little Germanic and tyrannical in that picture, and he was. He had a lab technician, Shelley and I were just talking about, named Barbara Thompson, who a few people knew, who had everything locked up and was not very supportive. But I was very fortunate in having come to what became a really great group of people a few years later, and you were introduced to all of them there, Larry and Palmer, Vince, and Paul. And in this picture, I think Steve, and because I want to be fair to him, looks more kind and more mellow, and he was in fact, but as Larry mentioned, he had some health problems, and because of that, he was literally unavailable and sort of unable to support me. And this is where Palmer really rescued me, and he became my guardian angel. I mean, he asked me to teach and promoted my teaching. He helped me with my research. He had me apply for, nominated me for grants. And then he encouraged me to obtain a tenure-track assistant professor position. I hadn't realized until Larry's list that there also weren't any women in the department then. And so he encouraged me to apply for the position. And then he announced actually what he's doing there, Larry, is I think he was actually announcing possibly that I would be an assistant professor at this FASEB meeting in 1980. So I subsequently moved through the ranks, as many of you know, and Palmer helped me all along the way. Something earlier than this that I didn't put on here is that he asked me to co-author a chapter with him in the Goodman and Gilman textbook. I was working on muscarinic receptors, and so I did that chapter along with him. He nominated me to serve as editor of the journal Molecular Pharmacology, which I did become. And then when he became the dean of the School of Pharmacy, he appointed me as interim chair of pharmacology. He was leaving to go to SSPPS, and he passed on the directorship of this T32 pharmacology training grant to me at the time that I was also selected as permanent chair of the department. He left me with an amazing faculty. You've seen lists of the faculty already. Here he is, my proud patron, Palmer. So he left me with an amazing faculty, the folks in red there, who we've all seen. And then he also had the wisdom to say that the research faculty who he hired in SSPBS should have appointments in a basic science department, many of them in pharmacology. So Sylvia Evans, Vivian Hook, Tracy Handel all became active members also of our department. And then for rather obscure reasons, Susan and Ed Dennis had their FTEs, which is your faculty money position, in the School of Medicine rather than in the department. And so I was active, and Palmer helped me to sort of arrest their FTEs out of the School of Medicine and into pharmacology, so they become bona fide, more than bona fide, larger than life members of the Department of Pharmacology. And then, so with the help of this really collegial and very, very strong faculty, I was able, and also the support of deans Ed Holmes and then particularly Dean Brenner, I was able to recruit a number of additional people over my 3 terms as chair, and they're all listed there. I don't need to read their names, and also to increase the number of FTEs in the department substantially. And Silvio Gutkind, who I hired in that 2015-2022 cadre of folks, has been selected and taken over from me as chair. So what I need to say is that none of the many milestones in my career would have happened. None of this or the many other milestones in my career would have happened without Palmer's tireless support and encouragement. He's been a colleague and a friend and a teacher for 50 years it is, and I'm enormously proud to be able to honor him today for all that he's done in his research, teaching, and training, in creating the school SSPPS, and in guiding this Department of Pharmacology to be what it is, the very best in the country. So thank you, Palmer, for, I could cry, but I won't, for all you've done for me and us.
Hardly stand up.
Are you going to say she's going to say
Oh.
just a word. I have no amazing. No slides. Okay. Welcome to this symposium honoring Palmer Taylor. I see so many friends and colleagues in the audience. I'm glad you could come in spite of all the forces that are at bay at this point. I'd like to applaud Palmer for the way he's chosen to run his laboratory, and especially for hiring me for the most fascinating job I could ever hope to have. The research end of Palmer's career is constantly evolving and changing. There have always been new projects and new people, yet he always maintains an atmosphere where science and investigation can thrive. He's always expected the best from people, and his guidance has produced some fine results. I've learned so much from you, Palmer, as well as from all of you who have come to honor him today. Thank you all.